Welcome to the ATAR Notes April lecture series. I'll be giving you the Head Start lecture for Biology, Unit 3 and 4. So hopefully everybody watching this is grade 12 or grade 11 doing either alternate sequence or accelerated subject. I hope your Easter holiday is going well, and thank you for attending these classes. It's just a way for us to give back to you, all of you senior students currently in grade 12. Just before I start today, I have a couple of things just to announce about ATAR Notes, the company. If you aren't familiar with us, we're basically a company that's created by past graduates for current graduates. So all of our resources are basically created by us and our community. And there are some really, really good quality free resources that you can get from us, including, the lectures that you're watching right now and the paid resources, which I will be talking about a little bit later. Um, while you're watching the lecture, I know it's not live streamed, but there will be a live Q&A session also answered by me. So if you've got any questions at all about QCEE biology in general or about any of the content that we're going through today, pop it in the chat and I'll give you guys a response. And please make use of that feature throughout the entire lecture. Today's lecture will be going for 2 hours, and you'll have the liberty of scrolling and cutting to all the places that you need to. But if you haven't done so already, please go to our website, atarnuts.com/register, and do create an account on there, just so you don't miss the next round of lectures, as well as all of the other free resources that are available on our website. So just because I've been talking about them, here actually are our free resources. We have heaps of free study notes and even some practice exams. I remember in grade 12, I managed to get a bio data test off the ATAR Notes website. Prior to actually working here, I was using the website as a resource for my own study. We've got our lecture series happening every single holiday. This one that you're tuning into is the April lecture series, and we do another one in June, September, and also for any grade 11s in the December holidays as well. We've got online Q&A forums that are monitored by our tutors. So if you have any questions throughout the semester about assessment types, study strategies, uni applications, if you guys are thinking about that right now, or whenever you do think about it, you can pop them into our little discussion forum and one of us will be able to give you an answer. We've got heaps of online revision videos up there for all of the QCE subjects. Daily newsletters, articles that are subject tailored and also tip tailored to provide you with the best advice that we possibly can. And also the one that you probably would have heard of already, the ATAR calculator, on there based on the last 3 years of results that are actually available to us. You can use that to sort of map out what kind of grades you want to be getting over the grade 12 just to achieve the target ATAR that you would like. And finally, these are our top tier resources. They get updated um, every year or so by our tutors, and these are the ones that you've got to pay for. So we've got TuteSmart, which is our tutoring program. That's online tutoring provided by recent grade 12 graduates. So I think everybody on the team right now has graduated from the ATAR system. Um, most of us being 2020 and 2021 graduates. We've got an array of study guides as well. I used to use these back when I was in grade 12. Unfortunately, I've given them to some family friends, but I had the biology course notes, the UCAT study guide, and the text guide. If you are either in English or literature and you struggle with analysis, or if you just want to get a second opinion about analysis, I strongly recommend getting those text guides. Half of the arguments I used in my external were like the based from analysis points in the text guide. They were so helpful. So I highly recommend that you check out the English study guides out for your particular book that you're studying. And finally, Ed Unlimited as well. So that is an online platform that allows you to have access to all of our study guides, not just for Queensland, but for New South Wales and what's the other? Victoria as well. So I didn't actually use that unlimited in grade 12, but I had it in grade 11 and I used it for practice exams and things like that because we didn't have many in grade 11 back when I was doing ATAR. Other than that, I would also like to say a huge thanks to our university sponsors, La Trobe, RMIT, Deakin, UTS, and Macquarie University. It's thanks to them that we're able to deliver lectures like these to you. So it's always good when we can do the holiday series because we get such a big level of engagement from you guys. And it's a chance to possibly make your life a little bit easier. So, just before we get into the content heavy portion of today's session, I do have a little introduction about me. So, hopefully you can see my little camera in the bottom right corner there. But hello, my name is Amandi. I graduated in 2021 from Ipswich Girls Grammar School. And I studied biology, chemistry, English specialist, methods, and Japanese. So I like to tell people that I did two of the sciences, two languages, and two maths subjects. Currently, I should say second year biomedicine student. Whoopsie. I'm a second year biomed student at UQ with provisional entry into medicine. So if any of you watching the lecture have any questions about med entry or just UQ uni life in general, pop your questions down below and I will be more than happy to answer. If anyone has any questions related to the content that we'll cover, my degree, or university life in general, please feel free to ask in the question box and I'll be monitoring that throughout the entire lecture as well. And the funniest thing is that I was once in your position back in grade 11 and 12 watching these holiday lectures on ATAR notes before I started working here as a tutor myself. So I find them very helpful in terms of getting head start on the content. I will say we do go through content pretty fast, so it's going to be quite a lengthy ride, but we've got a couple of topics to talk about today. I kind of want to cover the first half of Unit 1 in this lecture. So I've got DNA structure and replication. So what is DNA and why should we care about it? Cellular replication and variation. So this is us talking about mitosis, meiosis, and then how egg cells and sperm cells are created. As well as protein synthesis, which you might remember from grade 10, and gene regulation. If we have time, I can go through some general study tips, but this lecture is pretty packed. So throughout the lecture, I'll be using your input through the question box as well, so make sure to interact with that. All right, so most of the lecture will be just me talking at the screen like this. Sometimes I will flip my tablet around and write on the screen itself. But our first topic is just talking about what DNA is in general. So, DNA is the building block of all life. It forms what we call our genetic code, and it essentially has all of the instructions for how we appear, most of our behaviors, any adaptations, um, feedback responses, all encoded within these molecules. The actual DNA as a word is actually an acronym for deoxyribonucleic acid. And that basically means that it consists of nucleotides based on deoxyribose sugar. So you can see here, this is a strand of DNA that is sort of like a section of a strand of DNA. And you end up with three particular components per nucleotide. So nucleotide is like the base form of DNA. I'm just going to flip over my screen so I can annotate on the slides now. So these three components right here make up one nucleotide or one base component of DNA. We start off at the very top with something called a phosphate group. Connected to a sugar with 5 carbons in it. So this is our deoxyribose sugar. Connected to this sugar as well is our nitrogenous base.
that is one nucleotide. And the bonds between the phosphate group and the sugar are covalent bonds, which are very, very strong. And the bond between each of these, oh, sorry, never mind. There are four kinds of nitrogenous bases. We've got adenine, thymine, guanine, and cytosine. And adenine and thymine will always bind together. Guanine and cytosine will always bind together. Between the nitrogenous bases, connecting two strands of DNA together, we've got hydrogen bonds. And while hydrogen bonds are still quite strong, they can be easily broken by certain enzymes, which is going to be a pretty important component later on. DNA is a double-stranded molecule, meaning that it's got two parallel strands connected together, and they start to spiral together in a shape that we call a double helix, which is this structure that you see on the left-hand side there. In terms of the structure of DNA, it is all based around this thing called a pentose sugar. So pentose sugars are ones with 5 different types of carbon in them, all in one carbon chain. So we've got ribose sugar, which is the base form of RNA, which is another type of ribonucleic acid. We'll talk a little bit more about RNA later on, but deoxyribose is the sugar found in DNA. We've got carbon number 1 on the far most right-hand side there, carbon number 2, 3, 4, and 5. And then to this carbon chain, we've got two things that are being bonded to. And if you look on this side right here, you can see the phosphate group on the top will always be bonding to carbon number 5. So it'll kind of be coming... Oh, there we go. It'll kind of be coming off the side of carbon number 5, and then the other group, phosphate group, will be connected onto carbon number 3, and then it'll continue the chain on. So basically, we orient or talk about the structure of DNA in terms of its 5' end, as in the phosphate connected to the 5th carbon, and the 3' end, as in the phosphate connected to the 3rd carbon. The interesting thing, though, the parallel DNA strands that are bonded together by hydrogen bonds are actually anti-parallel or flipped over, essentially. So the 5' end of the strand on the left-hand side will only bond to the 3' end on the strand on the right-hand side. And as you go down, the 3' end on the left-hand side will bond to the 5' end on the right-hand side. So they're parallel to each other, but they're running in opposite directions, kind of like that. And that's important to know for your DNA replication section. So in terms of how DNA is stored within the cell, this is its structure in eukaryotic cells. So I'm just going to flip over again so I can write. Okay. So we end up with DNA as a double helix singular molecule here. And the strands of DNA start to wrap around and coil around these histone proteins, these circular ones right here. They join the histone proteins together kind of by loop around them like so, until you end up with a very long chain of chromatin. Each, as soon as a DNA strand will wrap around a histone protein, it is called a nucleosome when they're wrapped around together like this. Then chromatin will start to condense, so they'll start getting longer into these chromatin loops and squish together essentially, so they become more compact. And once chromatin becomes compact, it turns into a chromosome. Chromosomes have a couple of notable features. First is this center right here that connects the two chromosomes together. This is called a centromere. Here. And then finally, at the caps of the chromosomes, we have these structures called telomeres. Telomeres are caps that stop the DNA from unwinding back into chromatin, and centromeres are like sections of DNA that hold the two chromosomes together but don't actually code for anything. So this is our number one way that DNA is found in a cell, wrapped around histone proteins and condensed chromosomes. Histone proteins and chromosomes. Then we have our second way that DNA is found in the form of prokaryotic cells. So in the previous one, we were talking about eukaryotic or simple cells, sorry, more complex cells. These are our prokaryotes, so bacteria and things like that. There are actually two ways that DNA is found. Both of them are in the cytosol of the cell, if you want location. The first one is in the form of plasmids, which are these circular rings of DNA. And second way is unbound DNA found in the chromosome. Unbound DNA found in chromosome. So that's our second way. The QCEE actually has three different ways that DNA can occur, and this was a previous exam question. The first one, as we discussed previously, is in eukaryotic cells, wrapped around histone proteins. Histone protein in chromosomes. The second one is actually in the mitochondria. So every single eukaryotic cell will have mitochondria, which undergoes cellular respiration. They kind of look like this. And all mitochondrial DNA comes from your mother. So it's descended down the family without any influence from the sperm cell or from the father's genetics. This basically means that your mitochondrial DNA will be identical to your mother's, identical to your grandmother's, and this can actually be used to test for evolution or relatedness between organisms. So mitochondrial DNA, which is found unbound in organelles. The third way is as plasmids and unbound DNA in the prokaryotes. I strongly recommend that you note all of these three together. They came as a 2021 external paper question. And it was literally worded in the way that it was like, how does DNA occur in the cell? Which basically meant name the three ways that DNA is represented in the cell. And it got a couple people confused. So watch out for questions like that. All right, I realized that I need to do this again. So, our first big process that we're going to deal with in Unit 3 is DNA replication. Unit 3 actually deals with quite a few processes. So, our first one is DNA replication. We also cover mitosis and meiosis. We do protein synthesis, which I think is all we're going to cover in this lecture. But later on, you guys do some info on biotechnology practices as well that you need to know the steps for. So it's a whole lot of memorizing things and learning very, very lengthy processes. So DNA replication, I like to think of, has four different characters. We have, which are all different types of enzymes. The first one that we have is DNA helicase. The second that we have is DNA polymerase, which are our two most important ones. And these two are the only two that you really need to know a great detail for in the syllabus. The other ones that we have are primase and ligase. Now, while these two are not in the syllabus explicitly, I have seen multiple choice questions come up that deal with the roles of primase and ligase. So it's still important for you to know something about them. I don't think you need to explain their roles per se, but you need to know the process in terms of these four in these four enzymes. So our first step, we have our DNA double helix, kind of looks like this, right? Something like this. And helicase, which is our red enzyme, will come in and unzip the DNA helix, the DNA double helix, by breaking the hydrogen bonds, exposing the nitrogenous bases. So, as you can imagine, if those hydrogen bonds are broken between adenine and thymine and cytosine and guanine, we've got all of these bases which are now freely exposed to organelles, enzymes, ribosomes, anything that wants to do anything to them. So this is the way that your cell can access information that's coded within your DNA. Helicase will then continue to unwrap the DNA strand until it gets to the end of the gene. And then we move on to step 2. So step 2 starts to happen while helicase is unzipping, by the way. This is all happening at once. Primase will, will lay down RNA primus to the strands that are being copied. So essentially, if we're doing DNA replication, we want to have one DNA strand be copied into 2 identical DNA strands. So if this is my original strand of DNA, like so, and my helicase is continuing to unzip it, there's helicase there. Oh, I'm just gonna write all of the bases in like so. Primase will come in. What color did I make primase? Here we go. And basically lay down a primer at certain spots on the DNA strand that needs to be replicated. Kind of like if you were putting on primer before painting a wall, it's something that helps the paint adhere to the wall and get stuck on there properly. Or if you're doing like primer before putting on makeup, it's something that helps all of your makeup stick onto your face and stay. The primer for laid down by primase helps the copied DNA strand or the extra copy that we're going to make adhere onto the original copy more efficiently. So it's essentially like a priming step that helps the copied DNA strand stick onto the original DNA strand better. So now we go into step 3. Then comes in our DNA polymerase. DNA polymerase is also another enzyme, and it lays down complementary base pairs to the original or template strand. Pairs to the original strand. So this time I'm going to make this a little bit bigger, like so, like that. So imagine this is our 5' end of the original strand, and this is our 3' end. Remember, that's the end that the sugar is attached to, right? And we start off with a couple base pairs. Um, like so. And so we've got A, T, oh, oops. We've got A, T, T, A, G up here. So that means we have T, A, A, T, C on the top. What will happen is DNA polymerase will start coming in. So this is our polymerase. And joining up complementary base pairs to the template strand. So it'll say, okay, if this is a T, you get an A. This is an A, we get a T. We then get a T, another A, and then a G. And starts joining up all of the complementary nucleotides in one direction. However, the catch is that polymerase can only add bases or operate in the 5' to 3' direction. So here, if we've got our 3' end, it's starting off in the 5' end, right? And then moving this way. That's the only way that polymerase can continue working. So on this top strand, polymerase is all right to just continue, oh, whoops, sorry guys, continue working until the end of the strand and copy the same direction that helicase is unraveling in. However, for the bottom strand, technically, this starting point is the 3' end. Unfortunately, DNA polymerase cannot work in the opposite end, the 3' to 5' end. So it has to start somewhere random, essentially. So this is our polymerase molecule here. Sorry, wait. I'll make it start where I ended it before. So this is where polymerase will need to start, and then it can continue adding on the nucleotide base pairs in this order. So C, T, A, A, T. And then once it gets to the end of the strand, it has to jump back to a position it hasn't been to and continue replicating until it gets to the base pair before the base pair that it started on the previous time. So it would jump back until here and then continue building the strand until it got to this point. So because of this jumping back motion, we end up with a strand that has continuous replication, so it just follows the same direction as helicase, called the leading strand. And then a strand that has discontinuous replication or replication in certain chunks, called the lagging strand. However, polymerase will continue to add the nucleotide base pairs until we end up with two template strands that are completely separated from each other. So again, we've got 3', 5', 5', 3', and all the base pairs. Mixed with two completely copied or new strands fixed by DNA polymerase. Like so. The only problem is this strand on the bottom here will not be completely connected together. It'll be sort of in chunks. So as you can see here, there'll be some spaces between them. So this chunk will be separate depending on where DNA polymerase had to stop and start. This is where our final enzyme comes in. Here, this is ligase. Ligase is sort of like a glue enzyme. So ligase will glue all the fragments together, producing two identical strands of DNA. These individual fragments caused in the lagging strand are also called Okazaki fragments as well. That has come up on not a QCAA multiple choice question, but it came up in like a mock exam by a different company. So I'm just going to teach you guys, but they are called Okazaki fragments. Ligase's job is to come in and glue all of them together. And then finally, we end up with, hopefully this isn't over my video too much, but one of the DNA strands will be completely put together, and the other DNA strand will also be completely put together, and they will curve up into the double helix. So I've got a nice diagram of that on the next slide to show all of you. Here, this is a cleaner diagram, but it doesn't explain all of the steps. We've got helicase on this side. Wait, let me get the laser pointer. There we go. Helicase on this side unwrapping the DNA, breaks hydrogen bonds, exposes bases. That's what you've got to know. Then we've got primase coming down and laying down the RNA primers. We then have DNA polymerase, which is working to put all of the complementary base pairs together. You can see on the 3' end of the template strand, the 5' end of the copied strand is just being continuously made. And then on the 3' end, DNA polymerase is starting here and moving in the opposite direction. So that we get these little Okazaki fragments happening. DNA ligase then joins the Okazaki fragments together. So you end up with two parallel strands of DNA, and then they end up wrapping around each other to form two double helixes at the very end. Alrighty, so that's the whole process of replication, right? And replication is only one part of the cell cycle, which we're going to talk about now. And when we're talking about the cell cycle, there are 4 stages. Replication happens in the S stage, or yes, happens in the S stage, and it doesn't happen at random times. The S stage is essentially a preparation stage for cell division. Cell cycle kind of has 4 main stages. G1, which we see here, is the growth stage. So this is just where your cell is growing, maintaining itself, doing the function that it needs to do. And then it'll get to a point where it realizes that the organism is growing along with the cell and it needs to divide so that the organism can grow properly. Because obviously cell division makes more cells, so the organism gets bigger. Once the cell realizes that it needs to divide, it moves from the G1 phase, or the just stock standard growth phase, to the DNA synthesis phase. In this, DNA replication will occur. So you've got 2 copies of the DNA in the nucleus now. So the nucleus can divide into 2 daughter cells. Then we've got G2 phase, which is growth and preparation for mitosis. Luckily for you guys, you don't need to know any detail about the G2 stage at all. I learned all of that in uni. It is a pain. But basically, it's just where all of the checkboxes for mitosis will occur. And finally, the M phase is actually where the cell division happens. The nucleus splits, the cytoplasm splits, and you end up with 2 identical daughter cells from 1 parent cell. And that, oh, oops. And that process of mitosis is basically what we're going to talk about now. So, in your own time, I hope that you guys can pause and try some of these questions just to consolidate your understanding about DNA, because it's very important that
you're able to comprehend the information as well as absorb it. So have a go at doing those. But because it is a recording, I'm going to continue on with content. Now we're going to talk a little bit about cell variation and cell replication. So this is splitting of cells, cell division, and there are two types, mitosis and meiosis. Before we go on to talking about the actual processes behind those two though, I've got to talk a little bit about some basic knowledge. So first of all, we've got two types of cells, or two kind of categories of cells in our human body. The first one being somatic cells. So these ones are your regular body cells. They have no involvement in producing reproductive cells, and they've got nothing to do with fertilization and creation of gametes and zygotes and things like that. Germline cells, however, are the cells that end up producing sperm and eggs. So these two are the ones that fuse together to produce a baby in fertilization, basically. Diploid cells basically mean a cell that contains two sets of chromosomes. So all somatic cells have two sets of chromosomes, and we'll talk a little bit more about that in a later slide. Haploid cells, which are really only important for meiosis, refer to a cell only having one set of chromosomes, for example, the sperm and the egg cells. The reason being, sperm comes from your father, egg comes from your mother, and when the two of them fuse, you end up with a diploid cell, so the baby can have diploid cells. Um, so now we go into our two types of cell division. Mitosis is where a parent cell will divide into two genetically identical daughter cells. And then meiosis is a parent germ cell that divides into four genetically unique. Oh, no, that should be genetically unique. Haploid cells. So we're going to talk a little bit about that process very soon. So, in the last, in the earlier part of the lesson, we discussed how chromosomes are formed by condensing DNA wrapped around histone proteins, and we've got all of these. We've got 23 pairs of chromosomes within one cell, within one human cell. Every single one of our cells, no matter if it's a nerve cell, a muscle cell, a stem cell, a liver cell, will have this identical set of 23 chromosomes um, included. When you're doing DNA profiling, you'll end up with this structure or this diagram called a karyotype. And it's basically a layout of all of the chromosomes found within a particular organism or individual in this case, and ordered from the largest chromosome, which is usually chromosome number 1, all the way to the sex chromosomes, which are chromosome number 23. We have 22 autosomal chromosomes, so that means not relating to the sex chromosomes at all. And then one set of sex chromosomes, which determines whether you have male genitalia or female genitalia, or reproductive organs, I should say. XX is typically for male reproductive features, and XY is typically for female reproductive features. Later on in the unit, we're not going to cover it in this lesson, but you will be learning about sex chromosome mutations as well. So how intersex or individuals can originate and sort of what health implications you can have from there as well. So we've got each species will actually have its own set of chromosomes. In most humans, it is 26. However, chromosome mutations can cause different numbers of that to occur. And then when you're setting it all out in a karyotype, it's a paired set of 23 chromosomes. Our gametes are different in that they only have one half of the pair of chromosomes, which means that they only get 23. So later when we do meiosis and we have our egg cell and our sperm cell, each of those individual sex cells will only have 23 chromosomes. And so now I just want to go through the structure of a chromosome a little bit more, because this also came up as a exam question in my year, and it tricked people up a little bit. We have one chromosome here, and chromosomes in cell division will contain basically two copies of each other after the whole replication thing happens. So when they split, you end up with identical copies of each of the daughter cells. we've got two arms. Typically, a chromosome will have a short arm and a long arm, and a centromere at the center, which holds the two sister chromatids together. At the ends, we have these structures called telomeres, which are also made of DNA. They don't code for anything, they just stop the chromosome from unraveling and they help them stay in this like X shape. And then across the chromosomes, we've got all of these genes. And basically, the gene for one particular wait, the gene for a particular trait on one chromosome will be found in the same location in the other chromosome. So on these sister chromatids, we've got this could be like the gene for eye color. This is also the gene for eye color. This could be the gene for height. This could also be the gene for height, as examples. In reality, it is a lot more complicated than that, but typically speaking, genes are found on the same position and they are called the gene loci. So now that we've got all of that undone, our our next process is to talk about mitosis. Now, mitosis is by far the more basic version of cell division, and we start with this phase called interphase. So this is the base phase of the cell, and it's basically where the cell is growing, DNA replication has just occurred, and we have prepared for cell division. So the division step hasn't actually started yet. And then our first real step in the cell division process is that the chromosomes will start to condense down into the normal chromosome structure that we looked at before. Prior to that, they're not actually always in this X shape. They're kind of just floating around in the cell as these strings called, they're still chromosomes, but like they're still like floating around in the nucleus of the cell. Then, in prophase, the chromosomes will condense into the X shape and start to pair up with their sister chromatids. So we've got two identical chromosomes, four sister chromatids here. When the chromosomes are actually condensed like this, it's important to note the DNA can no longer be accessed. The telomeres have like zipped them all up, and the only thing that can happen from this point forward is cell division. So the DNA kind of goes into a more unwound state where it can be, oh, right here, never mind. And then the other thing that happens in prophase, along with the chromosomes condensing, is that the nuclear envelope, so the membrane of the nucleus, starts to degrade. And so the centrioles in the centrosomes right here will start to form spindle fibers. And we'll see that a lot later on in the latter steps. We then move on to metaphase. So this is where the spindle fibers coming from the centrosomes at the poles of the cell, which will hook onto each of the condensed chromosomes as they line up at the equator. So basically they hook up onto the chromosomes and pull them so that all of the chromosomes in the cell are lined up with the center of the cell. Think about it as metaphase, middle. They line up at the middle of the cell. Then what happens is in anaphase, the chromosomes which have lined up at the middle get pulled apart by the centromere. So that each of the sister chromatids gets pulled apart. And then in telophase and cytokinesis, all of the sister chromatids will reach the poles of the cell and they will start to form a nuclear envelope. So you end up with two separate nucleuses sharing one body of cytoplasm. And then in cytokinesis, the cytoplasm will split into two, and then the cellular membrane will form, making two identical daughter cells, which are diploid. That means that they've got two copies of each of the chromosomes. Right there. So, the more complicated step is our meiosis. And oops, sorry. This is our process of creating haploid daughter cells for the gametes. Again, we're starting with the same process in the sense that it follows interphase, prophase, metaphase, anaphase. But instead of having one round of that, you end up with two rounds of cell division. So in interphase, we start off with the chromosomes and the DNA being copied. This is a 2N cell as well, so we've got two copies of the same chromosome. In prophase 1, these chromosomes will pair up, essentially. So you've still got two chromosomes, but they've doubled, so you've got four sister chromatids in total. Metaphase then starts to occur, so the spindle fibers from the centrosome start to anchor onto the chromosomes and line them up at the equator. But instead of the chromosomes forming one underneath each other, homologous chromosomes, so chromosomes that are of the same size, will line up next to each other, and anaphase will pull apart an entire chromosome to the pole of the cell. One thing that's important to note here is that after metaphase 1, there is this process that occurs called crossing over. Oh, what is happening to the slides? Crossing over, which is basically where homologous chromosomes, so these are identical chromosomes that have been replicated. One is from the parent's side. So, sorry, that didn't make sense. One is from the father's side, one is from the mother's side, and their ends will start to overlap with each other and exchange sections of DNA. So this leg that is overlapping here, the overlapping section of the blue will belong now to the red chromosome, and the overlapping section of the red will now belong to the blue chromosome. These form something called recombinant chromatids, where basically the two copies of DNA have a combination of both the mother's DNA and the father's DNA on both chromatids. This is an adaptation that we've had that helps to improve our levels of genetic diversity because it shuffles the genes between the parents a lot more compared to if there wasn't any crossing over. The second thing that sort of happens in this section is independent or random fertilization, where each of these chromatids, whether it be a recombinant chromatid, one that has exchanged DNA with the other parent, or a non-recombinant chromatid, one that hasn't done the whole exchange, there's equal chance of these guys being fertilized. One isn't prioritized over the other. So that also helps to increase the level of genetic diversity in the gametes as well. So once crossing over has occurred and the chromosomes have shared genes with each other, anaphase starts to occur where the homologous chromosomes start to be separated to the pole of the cell. And then finally, the nucleus splits and the cytoplasm splits and the cellular membranes form to form two still diploid daughter cells. However, it doesn't stop there. We then go on to prophase 2. So we end up with two daughter cells, each with one version of the homologous chromosome in them. Metaphase 2 is where chromosomes will line up at the equator again and the spindle fibers will start to form. Then anaphase will pull each of these chromatids to this to the pole of the cell. So instead of having two copies of the chromosome like a normal mitotic cell division would have, this time we're only having one copy. So instead of 46, you only get 23 in the final one. Sister chromatids are then pulled apart. Telophase and cytokinesis occurs where the cytoplasm splits and the membranes formed. And we end up with four granddaughter cells that are all genetically unique. You can see, imagine if the darker one is the mother's DNA and the lighter one is the father's DNA. You've got one haploid daughter cell with entirety of mother's DNA, two which have combinations of both, and one with only the father's DNA. Each of them only having one copy of the chromosome, so it's a haploid or N cell instead of a 2N cell. Sorry, I went through that quite fast, but so much to go through with you guys. So we've already talked a little bit about crossing over, and finally, metaphase 2. At the end of meiosis, we end up with four granddaughter cells. However, there are two types of meiosis that can occur. Because at the end of the day, meiosis is making sex cells, right? So either our sperm cells or our egg cells. So there's slight variations in the production of sperm cells through spermatogenesis and egg cells or oogenesis, which is what we're going to be talking about now. Just checking on the time so that we're okay. So our first type of cell division is spermatogenesis. This is almost word for word recollection of the meiosis process. Our germline cells, which is our spermatogonium, or the original the origin point of spermatogenesis, will condense all of its chromosomes and copy them all so that they form a primary spermatocyte. This is where meiosis 1 happens and creates secondary spermatocytes. So instead of having two copies of every single chromosome, you end up with one copy of every single chromosome. Then meiosis 2 will occur and split all of those chromosomes up into individual chromatids, forming four identical sperm cells. Well, four sperm cells of the same size. They are all genetically different, though. And in terms of the timeline of this happening, after puberty, the spermatogonium forms the diploid primary spermatocyte through mitosis, which then undergoes meiosis 1 to form haploid secondary spermatocytes, and then meiosis 2 to form haploid spermatids. So these are our end cells at the very end. We then see that spermatogenesis is very closely following meiosis, forming two distinct, however, size identical sperm cells. These are then what fertilize the egg cell during fertilization and end up forming a zygote or fetus. Oogenesis, or the production of egg cells, is actually a lot more complicated. So before birth, all of the all of females will have a set number of eggs that are each 2n, called oogonium cells. And then through mitosis, all of their DNA will split into primary oocytes. However, this stays the same in prophase 1 until puberty hits and before the meiosis step can actually occur. So after puberty, you end up with meiosis 1 happening, which when it splits, so wait, let me go back. In meiosis 1, for spermatogenesis, we had two identical spermatocytes, right, in terms of size and structure. For meiosis, instead of them being identical in terms of size and structure, one of them will be bigger and one of them will be smaller. This is because at the end of oogenesis, we're not ending up with four viable egg cells. It's just not going to work that way. An egg cell is one of the biggest cells that you can have in your in the human body. And the fact that it becomes the basis for the zygote or the baby that comes afterwards means that it has to have a lot of cell mass for nutrients so that the organs can start developing, and a much larger cell mass full of nutrients and things that allow a cell or a baby to start developing. So instead of meiosis 1 resulting in two identical oocytes, we get one secondary oocyte and one polar body. This is straight after puberty. Then, after ovulation, meiosis 2 starts to occur, and you end up with. Oh, hold on. Then, after ovulation, hmm, hold on. Mitosis occurs before birth, then meiosis continues after, and you get a secondary oocyte and a polar body. After ovulation, sperm will start to enter the cell. It undergoes the second wave of meiosis, fertilizes, and you end up with a fertilized egg and a polar body. Oh, oh my God, what is happening? So, actually, let me go back to this. With oogenesis, instead of forming four viable gametes or four viable egg cells, you end up with one viable egg cell that has taken most of the mass of all of the others, and three polar bodies which eventually have to be discarded. One of the major differences between oogenesis and spermatogenesis is that spermatogenesis forms four the sperm cells of identical size and structure with different genetics, whereas oogenesis forms one egg cell with unique size and structure and three smaller polar bodies, which then have to be discarded by the body. So after cellular respiration, it's not respiration, cellular replication. We're going to talk a little bit about gene expression. So, as we talked a little bit about before, somatic cells and germline cells do not have the same function or appearance. However, through genetic study, we know that every single cell in a particular organism has or contains the exact same genetic code. And unfortunately, not all of that genetic code is going to be used by that cell because it has a certain structure that it needs to do. And to do that structure and function, they have to produce unique proteins. So, this process of protein creation is done through protein synthesis, which are broken up into two steps: translation and transcription. So those two together is the process that our cells use to turn the information encoded by our DNA into a usable form. For example, proteins and other functional molecules. Underneath the QCEE syllabus, we've got two important definitions that you guys need to know. I've copied these two directly from the QCEE glossary. And a gene will have regions of DNA that are made up of nucleotides. They are known as the molecular unit of heredity. And a genome is all of the genetic material in the chromosomes of an organism, including its genes and DNA sequences. These two I strongly recommend you memorize, along with any of the other definitions in the syllabus. They can come up as either short answer questions or multiple choice questions I have seen. So, within our DNA, I did touch on this a little bit earlier, but I'm going to go into detail now. We've got coding regions that actually code for a particular protein or are used as areas of DNA that create functional gene products. These are this actually makes up a very, very small part of the DNA. And you call these coding regions exons. So these are actually the ones that are useful in terms of creating proteins or gene products that end up doing a particular function for a cell. Every other aspect of the DNA is enclosed in a non-coding region. So these are regions of DNA that don't create proteins but are responsible for creating other structures like centromeres, telomeres, introns, which is the general term for non-coding regions, and important sections of regulatory genetic material, which are sections of DNA that we don't really understand the function of yet. We know that some of them form the centers of the chromosomes, some of them form the telomeres of the chromosomes, but in total, a lot of the functions of introns are not understood by humans right now. The most important function that we do know that they perform is the regulation of genetic material, and some of them can also exist as ribosomal RNA and transfer RNA as well. So after just before going into the actual process of DNA, we need to understand how DNA actually codes for proteins. So before I mentioned that each of the strands are organized in a series of base pairs in terms of thymine, adenine, cytosine, and guanine. However, in protein synthesis, we want to be talking about this U base instead of thymine, which is uracil. Cytosine, adenine, and guanine stay the exact same, fortunately. And each of these groups of DNA in the exons, remember the exons are the ones that are actually coding for proteins, will be read in groups of three. So each group of three can be read in the protein synthesis process, and they they produce one corresponding amino acid. And you'll be given this table in any exam questions relating to protein synthesis if you need to get the amino acid chain that is produced at the end of the process using this particular table right here. So I'm not going to dwell too much on this, but this is something that you guys will need to get acquainted to quite quickly. Alrighty, now we're actually going to go on to the process of protein synthesis. So I'm going to try and explain this
the best that I can. Just get my pen. Radio. So we've got our first step in protein synthesis, which is transcription. Now, the reason why this is called transcription is that this process stays in the same language. It stays in the language of nucleotide base pairs, language of nitrogenous bases. Oh, sorry. Bases. It occurs in the location where DNA is. So this is the nucleus of the cell. And essentially, the gene that needs to be encoded will be exposed. So for example, if this is the particular gene that we need to encode, it will be exposed and copied by an enzyme called RNA polymerase. RNA polymerase will get in here and copy the enzyme by attaching complementary base pairs. However, the important thing to note here is that instead of finding adenine from the original copy of DNA will bind to uracil in this strand. Now, this forms a single-stranded molecule of DNA, which is known as RNA, ribonucleic acid. Specifically, this happens because RNA has the ability to leave the nucleus. And that allows it to reach all of the structures that actually end up doing protein synthesis later on. So RNA has the ability to reach the nucleus. And this specific strand of RNA is called mRNA or messenger RNA. Think of it as containing a message that needs to get processed into a protein. However, fresh off the DNA transcription, this is still pre-mRNA because it hasn't been processed properly, but that'll happen in the next step. After transcription occurs, the mRNA molecule moves out of the nucleus into the cytoplasm for the next step. Moves out of the nucleus into cytoplasm. Okay. Then what happens is a series of steps called gene splicing. So this is what is called a post-strand transcription modification. If you're following along with the syllabus, they use this term, post-transcriptional modification. But basically, you end up with the original strand of pre-mRNA, which contains a mixture of both exons, which remember are our coding regions, and introns, which are our non-coding regions. Obviously, if you're trying to code a protein, you only want coding regions of DNA in there, otherwise you run the risk of mutation or the creation of a wrong protein. So gene splicing will occur where all of the introns are then cut out of the pre-mRNA strand and all of the exons are glued together, forming what is called a mature mRNA strand that is ready for protein synthesis and minimizes the risk of mutations occurring. After that, we end up in our final step, which is translation. So the mRNA strand is now brought into the cytoplasm of the cell, specifically in the area with ribosomes. ribosome rich area. So we end up with the mRNA strand right here. And then as it moves into the cytoplasm, a ribosome will come into contact with it. Imagine this is our ribosome. And the mRNA strand is read in groups of 3 called codons. Codons by structures called tRNA. So tRNA are these weird looking structures that kind of look like keyholes, like this, that are anchored onto by the ribosome. And they pair up the mRNA strand with a thing called an anticodon. So for example, if the mRNA original strand had a GAU code, the anticodon will pair it up with a CUA. The tRNA will then read the GAU codon and go, okay, GAU corresponds with, if I go back to the table from the start, I've got GAU ASP. So that's the amino acid that it needs to create. And then off the top of the tRNA, we will get an ASP amino acid. Then another tRNA molecule will move on to the next set of codons. So like so. So for example, if I had UAC, the anticodon will read AUG, and then UAC would form a TYR amino acid. So we still have our ASP. This orange one from before breaks off, hands the ASP to the next one, and then off the next anticodon will produce a TYR amino acid. This ribosome will then continue to form down the mRNA strand until it hits a stop codon. So if you look back onto the table from before, there are 3 stop codons, UAA, UAG, and UGA. You do not need to memorize this. If you are required a protein translation, you'll be given the table itself. But once the stop codon is hit, the ribosome and the tRNA will stop the translation process, and the chain of amino acids, which has been produced, also known as a polypeptide chain, the polypeptide chain will break off the tRNA and the ribosome. And condense into a 3D structure known as a protein. So essentially, this chain of amino acids will then bunch up into a 3D structure, kind of looks like a chicken nugget, and this is this is eventually the protein that ends up being produced. The reason why we call this translation is that we're going in the language of DNA, which is nitrogenous bases, into the language of proteins, amino acids. Because we're changing languages, it's translation. Language of amino acids. There we go.
So that is the process. I hope that makes sense. And here's another structure of it right here. You can see this teal colored structure is the ribosome, and it anchors onto the mRNA strand, which is like the pinky peach one that you can see here. The golden keyhole looking structures are your tRNA molecules, which form an anticodon to the codons from the mRNA. After reading the anticodon, it produces this amino acid, which are connected together by peptide bonds. If it gets to the stop codon, which are your UAA, UAG, and UGAs, we detach the polypeptide and it condenses into a protein. Just one other thing, the protein translation process will only start once an AUG codon has been read. This is our start codon, the red one down here, which produces the MET amino acid. And that's how the ribosome knows that, oh, this is the starting process, and it'll go down the chain of the mRNA until it hits the stop codon. So after that, um, oh, so after that, each of these codons will be read into an amino acid. So for example, AUG right here is our MET. I'm just going to translate it with you guys. The answer is on the next slide if you want to move forward, but I've got, oops. I have AUG, which is MET, ACG. So the way that you read it is you go down this line first. ACG, which is THR, UCA, which is serine, GAA, glue, AAA, lysine, AAU, ASN, CAA, GLN, CGG, ARG, GCA, ALA, GUG, Val, UCC, which is also serine, and then CCC, which is pro. So notice here we started off with a MET, so this is the start of the protein code. However, we didn't end up with any of the stop protein code. So this is only a fraction or part of a code for a protein. You'll only get a full protein code if it starts with MET and then ends with any of the stop codons. Like so. So here's the answer for it if you were watching the recording and you wanted to skip me going through it. But once again, this is only part of a code for a protein. In terms of what you would need to know, these are the syllabus dot points that I have transcribed over. So you need to know the difference between, or the definition of the term gene and genome. You need to understand that genes include coding exons and non-coding DNA, which have some functions like centromere formation, telomere formation, and introns, as well as functional RNA like tRNA, mRNA, and rRNA, which is ribosomal RNA. Recognize that many functions of non-coding DNA are actually yet to be determined. You will also need to know the process of protein synthesis in terms of transcription of a gene into messenger RNA in the nucleus, and then translation of mRNA into an amino acid sequence at the ribosome, and refer to transfer RNA codons and anticodons. So the way that I actually learned how to do this is I had like a whiteboard or a blank piece of paper, and I would draw out the process using diagrams, and every single time it would just go into my head much better than me trying to learn how to explain it using words. So now we actually, thankfully, we go on to our final portion of the lecture, and that is the importance of gene regulation. So I don't know why it's doing that. Gene regulation essentially refers to our ability to switch off genes and switch on genes depending on when we need to switch them on and off. It's a way of energy conservation because there are going to be times in your life cycle where you don't need the creation of certain proteins. So it would be a waste of energy and resources within the body to create proteins that you are not essentially needing. So gene regulation enables the turning on of genes and turning off of genes depending on when we need them or not. So this process of regulation starts very, very shortly after we've become like an egg cell. When the gene regulation starts to take hold of our cell and become more diverse with different functions as they form our body. This process is called morphogenesis. So, um, where you've got your egg cell and all of the you've got your egg cell which starts to divide into cells using the mitosis process. Each of these cells then needs to become specialized. So some of these cells will then grow on to become brain cells, liver cells, stem cells, muscle cells, whatever you need. That process of cell specialization is all controlled by gene regulation. So, mismanagement of regulation is essentially what causes cancers or other terrible pathologies, or conditions and things like that. If you think about two separate populations of cells, so for example, in this little diagram here, we've got a stomach cell, and it's got a stomach cell ending up with hydrochloric acid. So hydrochloric acid is one of our stomach acids that helps digest food. So this eye cell has just created a protein that helps with the production of hydrochloric acid. So the eye cell department is completely producing hydrochloric acid when this is supposed to be for the stomach cell. The eye cell gets confused because it doesn't think that it's on the stomach and ends up burning the eyes of the human. So gene regulation is really important so it doesn't create proteins that don't need to be in locations that they don't need to be in. Um, so for example, we've got stomach epithelial cells that create gastric acid, so like the ones in our little picture there, and cone cells in the retina of our eyes that are responsible for us being able to see color. If for some reason the regulation of genes in the cone cell failed, it would become the cancer of the eye, or uncontrolled protein creation in the eye. Another possibility would be that the cone cell starts to downregulate its normal cone cell genes, so the genes that are able to see color, and upregulate or stimulate the genes that are found in stomach cells for some reason. And therefore, the cone cell will start to transform into a stomach cell, doing all of the normal jobs that an epithelial cell would do, secreting gastric acid and absorbing nutrients. And this is a huge problem because obviously we don't want anything that the stomach does to be happening in our eyes. So I mentioned two words previously, one of them called upregulation and the other one called downregulation. These are both determined by something called the activator and the repressor. So next, these activation and repressor functions are carried out during the transcription stage of protein synthesis. So transcription actually ends up being a pretty key regulatory point for all of our genes. There are things called transcription factor proteins. So these are proteins that specifically stimulate the transcription of a gene or downregulate or inhibit the transcription of a gene. They will bind to specific DNA sequences in or near a gene and either stimulate or repress the transcription into RNA. Basically, they bind to parts of DNA that stop specific genes from being transcribed or increase the rate that we're being transcribed. So if I get the laser pointer back up, we've got this pink protein binding onto a site. This is a repressor protein, right? So it actually stops all of the mRNA from being accessed and it doesn't result in the transcription of the target gene. The repressor blocks the general transcription factors and RNA polymerase from actually hooking itself onto the DNA strand. So no transcription can occur and this protein will not be expressed. On the other side, we've got activators. So these are like our green sites here. It will activate onto a binding site that hooks RNA polymerase onto the DNA strand itself, which lets the RNA polymerase transcribe the target gene, which is in purple over here. As these transcription factors are also proteins encoded by the DNA, they can actually have transcription factors that regulate them as well. So it becomes this like pathway of proteins influencing genes. However, that's a little bit more complicated than you guys need to know. The genes that encode for a transcription factor are called regulatory genes, as they are encoding for these regulatory factors which control the expression of functional genes.
When we talked about growth factors before, um, and sorry, when we talked before about growth factors, these actually act via a very similar mechanism, where their signaling pathways cause gene expression to be promoted or inhibited by signaling the action of activator or repressor proteins. So there are a couple of different kinds of gene regulation that QCAA expects you to know. The first one is about how sex is determined in humans using something called the SRY gene or the sex gene. Most of your chromosomes come in homologous pairs. So these are identical pairs that we talked about in the um, mitosis meiosis, cellular replication step. The two chromosomes of a homologous pair contain the same basic information. That is, the same pair of genes in the same order organized across the chromosome. But many carry different versions of those genes. So if any of you guys have done grade 10, grade 10 biology, you would know about dominant and recessive genes. They can carry sorry. These chromosomes can carry the same kind of gene, but one chromosome may carry the dominant version of the gene or the dominant allele, and the second version of the gene may carry the recessive allele. So there is only one set of chromosomes that isn't arranged in a homologous pair, and that is your sex chromosomes. So for people who are XX, so typically with female reproductive organs, they have the opportunity to have homologous chromosomes because they're the same size. So each of these genes will be organized in the same way. Males, on the other hand, with XY chromosomes, will have different sized sex chromosomes because the Y chromosome is a lot smaller than the X. Um, and they aren't considered homologous. So instead of an X or and a Y, a human female will have two X chromosomes. Because X chromosomes do actually form a homologous pair, these genes can actually show, sorry, because X chromosomes do not always come in homologous pairs, these genes they carry show unique and distinctive patterns of inheritance. So again, if you guys remember doing things with pedigree charts, you would have known that there would be like sex-linked genes, where the chance of you getting this particular mutation would be higher depending on whether you had whether you were a male or a female. So human X and Y chromosomes determine the biological sex of a person, with XX specifying female and XY specifying male. Although the Y chromosome contains a small region of similarity to the X chromosome, here you can see that they have both of them have at least one of the X chromosomes, and the Y contains some similarity to the X chromosome. The Y chromosome is much shorter and contains way fewer genes. To put some numbers to it, the X chromosome has about 800 to 900 protein coding genes with a really, really wide variety of functions, while the Y chromosome only has 60 to 70 protein coding genes, about half of which are active only in sperm producing organs or in the testicles. The human Y chromosome plays a key role in determining the sex of a developing embryo. This is mostly due to a gene called SRY, or the sex determining region of the Y gene. SRY is found on the Y chromosome and encodes a protein that turns on the other genes required for male development. XX embryos typically do not have an SRY. So this is our typical female chromosome orientation there. They do not have the SRY gene. So they will develop as female, and all of the genes that stimulate female reproductive organs and female sex characteristics will start to be switched on. If an SRY bearing X chromosome fertilizes a normal egg, it will produce an atypical or chromosomally an atypical female embryo. So it'll still have an XX chromosome combination, but because it's got the SRY gene, this individual will start forming male sex characteristics. Um, so this is a instance of intersex humans developing, basically. If an SRY deficient Y chromosome, so this is our XY
this is an individual with an XY combination, but it doesn't have the SRI gene, it won't start producing male characteristics. Instead, it'll be a female with XY chromosomes. Another an example of intersex. So if you want to think about it simply, the default sex characteristic will always be female sex characteristics because most of the mass that you're getting from the zygote is from the female egg cell. Male sex characteristics will only start to develop if the SRI gene is present. And if it's present in where it shouldn't be present, a male with male sex characteristics will only have an XX chromosome combination, and a female without the SRI gene will have an XY chromosome combination. So there, in terms of what the SRY gene is actually doing, it encodes a protein called the SRY protein, which is a regulatory protein. It acts as a transcription factor, which causes the upregulation of other transcription factors. One important transcription factor that it upregulates is the SOX9. Sorry, just give me a second. SOX9, which is responsible for turning a population of undifferentiated cells into specialized cells, which go on to forming the testes in males. It also helps with the development and maturation of the testes and male reproductive organs, which eventually go on to secrete testosterone and other male hormones, which eventually develop male sex characteristics. If this gene is not present, so if there is no SRY gene, you end up with no production of testosterone and no production of male sex characteristics. So by default, even if they've got XY chromosomes, the human that develops will have female sex characteristics and female anatomy. So the next type of transcription factor that the QCEE wants you to have a look at is called Hox genes or homeobox genes. These genes are controlling the structures of the body or like sections of the physical body. And when misexpressed, it can cause some really, really crazy mutations. Now, the main model of Hox genes, when they're usually studied, are a fruit fly. And typically, a fruit fly will have 6 legs. These legs usually grow out of the thorax, as you can see labeled B. So all 3 of these appendages are coming out of this midsection here, the thorax. And it is the Hox gene that is responsible for the development of these structures in the correct location. There are other regulatory genes that are involved, but they're all sort of controlled by the Hox gene. The genes that are determining the identity of particular segments, as in what ends up growing as the head, what ends up growing as the thorax, and what ends up growing as the abdomen, are all dictate what cells go into which region of the body. So if you consider a particular type of Hox gene, this a Hox gene in the fly, it is responsible for the formation of the thorax of the insect. The gene here acts as a master regulator and activates all of the genes responsible for creating this fly's second pair of legs and the other structures specific to the second thorax segment. If the gene is not correctly expressed here, we get a normal, if the gene is correctly expressed here, we get a normal looking fly with 3 sets of legs coming out of the thorax. However, if this gene is expressed in the wrong section, which, happens after a mutation, some pretty strange things can happen. Say, for example, this fly had a mutation and it caused the Hox gene to grow in the fly's head or be expressed in the fly's head instead of the antenna. The gene activating the program would typically develop the second segment of the thorax and its associated features in the wrong place entirely. So what you essentially see there is the antenna of the fly, antenna of the fly growing out of the head, growing as legs instead. So mutations in the Hox gene cause abnormal structures to form in different segments of the body, basically. Alrighty. Do I want to keep my incantations? Yes, I do. Just before we finish off today, I would like to show you some features of protein synthesis. I have a couple videos to show you guys as well. Oh, here we go. So I don't know if sound is going to be transmitted, so essentially I'm going to just speak over it. But with protein synthesis, you end up with the larger cell here. Within the cell, we enter the nucleus. And so our nucleus, we enter our chromosome. So this is all of our condensed DNA. And wrapped around histone proteins, and at its core, we've got a double helix which codes for a gene. So now we've got RNA polymerase here. RNA polymerase will start to connect all of the complementary bases of the particular gene to each other, replacing adenine with uracil. As it's complementing, the strand that is being formed is an mRNA strand. And so this mRNA strand will start to move out of the nucleus into the cytoplasm where it can reach a ribosome. I'm just going to pause here. The animation doesn't show it, but during this step is when gene splicing will occur, and all of the introns will be removed and the exons will be spliced together so that the mature mRNA strand is created. And here we have it attaching to a ribosome. Within the ribosome, you get these keyhole structures that kind of look, keyhole structures called tRNA, which have an amino acid attached to them and will latch onto the corresponding codon to its anticodon. So here, the anticodons will latch onto the codons from the mRNA, producing a polypeptide chain formed together by peptide bonds, by the way. That is what connects them together. And then at the end, the polypeptide chain will detach from all of the tRNA once the stop codon has been hit, and then condense to form a protein, like so. This protein will then go on to do, it can go on to stimulate hormones, it can go on to catalyze reactions, it can go on and be an enzyme of some sort, we don't know, but at the end of the day, that's the formation of a functional gene product. I also have a video to show you guys on cellular replication as well, DNA replication. That will hopefully make a bit more sense as well. Here we go. I think it's the same people making these videos, but DNA replication starts to unwind the double helix, and then helicase will start to come in and unwrap the strand. Sorry, here they're just talking about how adenine bonds to thymine, cytosine bonds to guanine. So we have 5' end, 3' end, depending on what sugar the phosphate group is bonded to. So here we have helicase coming in, breaking the hydrogen bonds and exposing the bases in something called a replication fork. Then we have primase. So primase forms RNA primers basically that help the strand latch on to the, help the copied strand latch on to the template strand. DNA polymerase will then come in and start to copy the template strand by attaching nucleotide bases. Just as a note, by the way, nucleotide bases are added on, they kind of like freely float around the cytoplasm, if you're wondering where they come from. Um, it's, and DNA polymerase in DNA replication and RNA polymerase in protein synthesis kind of have the job of rounding up all of these bases and using them to um, complement the template strand. So DNA polymerase is here, joining up all of its uh, base pairs, and it works in the 5' end through to the 3' direction. So this is our leading strand. It's nice and continuous here. And it'll stop until it gets to the end of the DNA strand, like so. On the bottom there, we've got a lagging strand. So it starts halfway through the strand that's been unraveled and starts continuing on in the 5' to 3' direction, forming an Okazaki fragment. And basically, DNA polymerase will jump back. Well, that's primase creating another primer. DNA polymerase will then jump back and start to put the DNA bases in order between the newest primer and the section that it made before. And it'll repeat the process. So primase comes in, forms its primer, polymerase will come in and fill in the blanks. Like so. So you end up with all of these individual pieces. Exonuclease is something that you don't need to know about, but essentially it gets rid of the primer and DNA polymerase can come in and replace everything with DNA. So after that, DNA ligase will come in and seal all of the bonds between the leading strand and the lacking strand. So it connects together all of the Okazaki fragments. And once completed, the two identical DNA molecules will spiral into a double helix again, forming a new strand and a copied strand. And that's it. Okay, so now I actually have some questions for you guys to do. Or for me to go through with you, I should say, because we're not doing any. Oh, go back to OBS. I want to give you guys a small break just to get refreshed and everything, and then we'll go through some practice questions together. Mm, okay, I'll come back in 2 minutes. Okay, so hopefully you guys were able to take a little break. Obviously, pause for more than you need. For more time than you need if it is a recording at the end of the day. Um, and I have some practice questions to go through with you guys now. So basically,
I'll give you my approach of how I would do it in uh exam situation, but I've got a couple of multiple choice questions to start off with. A couple of questions on protein synthesis and DNA replication. I think there's a couple of them in there as well. So, first of all, when I'm coming through a multiple choice question like this, I kind of want to look at all of the answers before choosing an answer. So I've got a genome is defined as a genome, remember, is all of the varieties of genes within a particular organism or the combination of all genetic material in an organism. So by far the option that matches that the most is option B. The molecular unit of heredity, our first one there, is more a gene than a genome. And the sequence of triplets and DNA nucleotides that make up a gene are codons. The combination of alleles for a particular trait carried by an individual, this one isn't actually one that we talked about in this lesson, but this is a genotype, which might be revision from grade 10 for you, I'm thinking. Um, and then question 13, which of the following molecules is involved in the transcription of DNA? Remember, transcription was the process where the DNA coding strand is, oh, yeah. So our DNA coding strand is transcribed into messenger RNA. And there's only one important enzyme for you to know in this process, and that is RNA polymerase. So RNA polymerase is our answer. DNA helicase is used in DNA replication. Transfer RNA, or tRNA, is used in the translation step of protein synthesis, so not transcription. And amino acids are production of translation, or a product of translation. So our obvious answer there is RNA polymerase. Next, we have a phenotypic expression of genes is regulated by what? Now, immediately whenever you see regulation and genes being used, you have to think of transcription factors immediately. So remember, some transcription factors will upregulate or stimulate a gene, and other transcription factors will downregulate. Random fertilization is used to promote genetic diversity and meiosis. DNA polymerase is used in DNA replication, and so is helicase. So the only thing that is relating to gene regulation is transcription factors. What is the role of DNA polymerase in DNA replication? Here we go. So DNA polymerase is our third enzyme that comes into play, which polymerizes the new strand of DNA. So they add complementary nucleotide bases to the exposed DNA strands. That is answer B. These are questions from the 2022 paper, so the one that just came out. By the way, you can find all of these questions on the QCAA website with marking guides attached if you still wish to check them out after today's lecture. What is the molecular unit of heredity? Well, that is a gene, nice and easy. Question 20. The section of DNA is made up of two strands, 1 and 2. Now, here, if adenine binds to thymine, oops, adenine will bind to thymine, you would expect equal amounts of adenine and thymine on the opposite ends of the DNA strand. So this is strand 1, this is strand 2. If I have the thymine here, it's going to be bonded to an adenine on the top half. So therefore, if I've got 29% thymine on strand 1, that means 29% of adenine will be featured on strand 2. Similarly, 25% of cytosine will be corresponding with the same amount of guanine on the other side. So guanine on strand 1 will be 25%. 14% of cytosine corresponds with 14% of cytosine on strand 1. Therefore, to find out the remaining, you just have to take all of those numbers away from 100. So what, 29 over 100 minus 29 minus 25 minus 14 is 32%. Oh, why is that? And 32%. So it can be inferred that strand 1 contains 25% cytosine. Well, no. Strand 1 contains 14% cytosine. Strand 1 contains 32% adenine. That sounds right to me. I'm just going to pick it, but I'll check through the others just in case. Strand 2 contains 29% thymine. Well, no, that's wrong. And then strand 2 contains 71% adenine. Immediately, I know that's like false. So our correct answer is B. Next question. So this was a question that came up in 2020 paper 2. Remember, you're wanting 4 marks here, so they're expecting us to use 4 pieces of information for each of these 4 marks. The verb that they're using is explain, so they're expecting us to put in detail. We're not just identifying or describing a process here. We're explaining the process of protein synthesis in terms of transcription and translation. So, transcription is our first step. In the nucleus of the cell, mRNA is, oh wait, no, I should start it with the DNA sequence of the target gene the DNA sequence of the target gene is transcribed into a single strand mRNA molecule by RNA polymerase. And then for translation, the mRNA strand is contacted by a and tRNA will oh, oops. tRNA will read the mRNA strand in codons. Yes, keep them. Codons producing a corresponding amino acid. Once, from start codon to stop codon. Complete. The amino acid chain, polypeptide chain, will break off and condense into protein. Like so. So that's what an example of what I would write. This is the official marking guide that they've given. So transcription involves copying a gene's DNA sequence to make an RNA molecule. This is performed by uh, enzymes called RNA polymerases, which link nucleotides to form an mRNA strand. In the translation process, the mRNA formed in transcription is transported out of the nucleus into the ribosome. Here it directs protein synthesis, and the mRNA passes through the ribosome and the tRNA interacts with it, adding amino acids to form a protein chain. When you actually look at the marking guide, it's kind of stupid, honestly, but DNA is copied, you get 1 mark for saying that. mRNA is formed, you get another mark for saying that. You get 1 mark for saying that mRNA is transported to a ribosome, and then tRNA adds amino acids to form a protein chain. So as long as you got those 4 pieces of information in your response somewhere, you should be all good to go in terms of your response. Okay. For this one, if you want, pause the recording and see if you can try this one by yourself. But this is an example of you taking a template DNA strand and then finding out what proteins it's going to code. So pause here, try it out, and then I'm going to go ahead and do myself. So all of our T's turn into A's, C's turn into G's, G's turn into C's, G's, G's turn into C's. So we've got C, G, C, G. A's will turn into U's, remember, for mRNA. C, U, C, G, A, U, C. The anticodon for the tRNA is the opposite mRNA opposite mRNA strands for each codon. So that becomes U, A, G, C, G, C, A, G, A, G, C, U, A, G. However, each of the amino acids that is formed is from the original mRNA strand. So I'm reading AGG, which is OG, CGC, which is also OG, GUC, which is Val, UCG, which is serine, and AUC, which is ILE, like so. And so that's the model answer. That would be an example of what you're required to do. Then I've got another one for you guys to practice very, very quickly. mRNA here is made during the transcription process, and mRNA is made in the nucleus of the cell. So once again, same thing. A turns into U, C turns into G, G turns into C, U, G, C, C, A, U, G, T. Turns into A, G turns into C, and A turns into U. So this becomes A, C, A, G, A, C, G, G, U, A, C, U, G, A. And then we read the mRNA off the table. So I've got UGU, cis, UUC. Oh, wait, no, UCU, sorry. Serine. GCC. Hello. AUG. Met. So technically speaking, this is the start codon, and this is where the translation would start. and then ACUTHR, like so. Um, I just got a couple of base questions for you. What is the first step? Please pause the recording or try and do these questions yourself. Oopsie. What is the first step of protein synthesis? That is transcription. Oops. Second step is translation. Where does the first step of protein synthesis occur? In the nucleus. Where does the second step occur? Cytoplasm. But more specifically, with the ribosomes. Nitrogen bases are read 3 bases at a time, and the bases on the mRNA strand are called the codons. The bases on the tRNA strand are called the anticodons. Start codon produces met, and it is AUG. This is the only one that I think you need to know. The stop codons, read them off the chart. They are UGA, UAA, and UAG. So UGA, UAA, and UAG. A bunch of amino attached together is called a polypeptide chain. Oh, sorry. Alrighty. So, hopefully that wasn't too bad. Hopefully that all made sense. Gonna go back here. As always, if you guys have any questions, please put them into the chat. I will be staying back for a little while as well to answer any questions that you may have. Lastly, just before I finish, I'm just gonna get back up this and hopefully you guys can see that. Please make sure to check out the rest of the lectures in the series if you did like this lecture. I know that there's a lot of other lectures going forward just on like the QCE system in general, as well as the other main subjects that we've got. Please register. You'll be able to access the recordings and the live chat to them as well if you get there in time. Other than that,